In this video, we are going to learn about the radiation dosimetry and its working and its parts, application and importance. Radiation dosimetry is the process of measuring and quantifying the amount of ionizing radiation that a person or object has been exposed to. This information is important for determining the potential health hazards associated with exposure to radiation and for establishing safety protocols in radiation-related industries. Here is a detailed essay on the working, parts, application and importance of radiation dosimetry and working of radiation dosimetry. Radiation dosimetry involves the use of various instruments and methods to measure the amount of radiation exposure. The main components of a dosimeter include radiation detector. This is a part of the dosimeter that detects and measures the amount of radiation exposure. Different types of detectors are used depending on the type and energy of the radiation being measured. Different types of detectors are used depending on the type and energy of the radiation being measured. Then, readout device. This is a device that displays the dose measurements from the detector. It can be a digital display or a film that is developed after exposure. Then, a power source is needed. This provides the energy needed to operate the detector and readout device. The working of a dosimeter involves exposing the detector to ionizing radiation, which causes it to produce a signal that is recorded by readout device. The signal can then be used to determine the amount of radiation exposure. Parts of radiation dosimetry. Radiation dosimeter can be divided into two main categories: personal dosimeter and area dosimeter. Personal dosimeters are worn by individuals who work with ionizing radiation to monitor their exposure. They can be in the form of badges, wristwatches, or wristbands or rings, and contain a small amount of radiation detector that can be read periodically to assess the dose. Area dosimeters are used to measure the radiation exposure in a specific location, such as a room or facility. They are typically larger and more complex than personal dosimeters and may include multiple detectors to measure different types of radiation. Now, what are the applications of radiation dosimetry? Radiation dosimetry has many applications in various industries, including medical imaging. Radiation dosimeters are used to measure the amount of radiation exposure during medical procedures such as X-rays, CT scans, and nuclear medicine exams. Then the radiation therapy. Dosimeters are used to measure the radiation dose delivered to cancer patients during radiation therapy. Nuclear power plants. Dosimeters are used to monitor the radiation exposure of workers in nuclear power plants to ensure their safety. Radiation research. Dosimeters are used to ensure the radiation dose in experiments involving ionizing radiation. What is the importance of radiation dosimetry? Radiation dosimetry is important for several reasons, like it allows us to determine the potential health hazards associated with radiation exposure and establish safety protocols to minimize these risks. It ensures that radiation exposure is within safe limits and prevents unnecessary harm to individuals and the environment. It allows for accurate and efficient monitoring of radiation exposure in various industries like the medical imaging and nuclear power plants. In conclusion, radiation dosimetry is an important field that allows us to measure and quantify radiation exposure. The different parts of dosimetry instruments and methods of radiation detection play a crucial role in the accuracy of dose measurement. Accurate and reliable radiation dosimetry is essential for ensuring the safety of individuals and the environment in industries that involve ionizing radiation. Now let's learn about the various radiation measuring counters like GM counter, solid scintillation counter, liquid scintillation counter, proportional counter, semiconductor detectors, and they are working along with their application. Radiation detection is a vital aspect of various fields including healthcare, nuclear research, and energy production. Different types of radiation detectors are used for different applications. Here are the explanations of some of the commonly used radiation detectors, including their parts, working, and applications. First is the GM counter, also called as a Ziegler-Muller counter, is a gas-filled radiation detector that detects alpha, beta, and gamma radiation. The GM counter is a gas-filled detector, and the GM counter consists of a metal tube filled with a low-pressure gas. usually argon or helium, and a thin wire at the center of the tube. The tube is surrounded by a high voltage power supply and the wire is connected to a counting device. So what is the setup? There is a chamber and a metal tube filled with a low pressure gas, usually argon or helium. A metal tube filled with gas is there. That gas is of low pressure and that is probably argon or helium, and a thin wire at the center of the tube will be there. So tube is filled with the low pressure gas like argon or helium. There is a metal tube filled with low pressure gas and a thin wire at the center of the tube is attached. This tube is surrounded by a high voltage power supply and the wire is connected to a counting device. When ionizing radiation enters the tube, it ionizes the gas molecules which cause the electrons to move towards the wire. This generates a current pulse that is detected by the counting device. So that is how it works. The Giga-Muller counter, GM counter works. There is a metal tube filled with low pressure gas like argon or helium, and there is a thin wire at the center of the tube, and this tube is surrounded by a high voltage power supply. And when ionizing radiation enters the tube, it ionizes the gas molecule which causes the electrons to move towards the wire. This generates a current pulse that is detected by the counting device. Now let's learn about the applications. GM counters are used for detecting and measuring radioactivity in medical applications, environmental monitoring and nuclear power plants. Which are the fields where GM counters used? Medical applications, environmental monitoring and nuclear power plants are these major sites. Next is the solid scintillation counter. A solid scintillation counter is a detector that uses a crystal scintillator to detect ionizing radiation. The scintillation emits light when it interacts with ionizing radiation. A solid scintillation counter consists of a scintillation crystal, a photomultiplier tube or PMT photomultiplier tube, and associated electronic components. Working. When ionizing radiation interacts with the scintillator crystal, it produces light photons that are detected by the photomultiplier tube. The photomultiplier tube amplifies the signal and sends it to the electronic components for processing. So photomultiplier tube is very important. Solid scintillation counters are used in nuclear medicine for detecting gamma rays emitted by radioactive isotopes. Next is the liquid scintillation counter. A liquid scintillation counter is similar to a solid scintillation counter except that it uses a liquid scintillator instead of a crystal scintillator. A liquid scintillation counter consists of a vial containing a liquid scintillator, a photomultiplier tube and electronic components. When ionizing radiation interacts with the liquid scintillator, it produces light photons that are detected by the photomultiplier tube. The photomultiplier tube amplifies the signal and sends it to the electric components of the system for processing. Which are the applications? Liquid scintillation counters are used for detecting low energy beta particles emitted by radioactive isotopes in environmental monitoring and nuclear medicine. Earlier, we have learned about this solid scintillation counter that was used for detecting gamma rays. Here we are detecting weak beta particles, low energy beta particles. Earlier we were detecting about gamma, now we are detecting beta. Now the proportional counter. A proportional counter is a gas-filled radiation detector that detects alpha, beta and gamma radiation. It is similar to GM counter, but it operates at a higher voltage and detects a wider range of radiation. Which are the parts? The proportional counter consists of a metal tube filled with a gas, usually argon or helium, a wire at the center of the tube and electronic components. When ionizing radiation enters the tube, it ionizes the gas molecules which causes the electrons to move towards the wire. What is happening when ionizing radiation enters the tube? It ionizes the gas molecule which causes the electrons to move towards the wire. The proportional counter detects the signal produced by each electron, amplifies it and sends it to the electronic components for processing. Which are the applications? Proportional counters are used for detecting and measuring radioactivity in medical applications, environmental monitoring and nuclear power plants. Next is the semiconductor detectors. Semiconductor detectors are solid state detectors that use semiconductor materials such as silicon or germanium to detect ionizing radiation. The semiconductor detector consists of a semiconductor crystal, electrodes will be there, and electronic components, associated electronic components. Then let's learn about the working. When ionizing radiation enters the semiconductor crystal, it creates electron-hole pairs. We are going to learn about the semiconductor detector. So semiconductors are middle level in between insulators and conductors. When ionizing radiation enters the semiconductor crystal, it creates electron-hole pairs which generate an electrical signal. The electrodes which generate an electrical signal, the electrodes detect the signal and send it to electronic components for processing. What are the applications? Semiconductor detectors are used in medical imaging and nuclear research for detecting gamma rays emitted by radioactive isotopes. മെഡിക്കൽ ഇമേജിങ്ങിലും അതുപോലെ ന്യൂക്ലിയർ റിസർച്ചിൻ്റെ ഭാഗത്തും ഇത് ഉപയോഗിക്കുന്നു. Semiconductor detectors are used in medical imaging and nuclear research for detecting gamma rays emitted by radioactive isotopes. So in conclusion, different types of radiation detectors are used for different applications, and the choice of this one is depend on the person or the requirements. Autoradiography types, principle, parts and working, and applications and importance. Autoradiography is a technique that is used to visualize the distribution of radioactive isotopes in biological samples. So that is what autobiography is done for. Autoradiography is a technique that is used to visualize the distribution of radioactive isotopes in biological samples. This technique is commonly used in molecular biology, biochemistry and medical research. There are two types of autoradiography: emulsion autoradiography and film autoradiography. So let's learn about the emulsion autoradiography. Emulsion autoradiography is based on the principle that when a radioactive isotope decays in a biological sample, it emits radiation that interacts with the photographic emulsion. The interaction results in the formation of silver grains in the emulsion, which can be visualized under a microscope. So that is the principle of emulsion autoradiography. Emulsion autoradiography is based on the principle that when a radioactive isotope decays in a biological sample, it emits radiation that will interact with the photographic emulsion, and silver grains are formed in the emulsion, which is visualized under a microscope. So parts and working. To perform emulsion autoradiography, a thin layer of photographic emulsion is applied to the surface of the biological sample. The sample is then exposed to a radioactive isotope which decays and emits radiation. The emitted radiation interacts with the emulsion, resulting in the formation of silver grains. After the exposure, the sample is developed, fixed and stained to visualize the silver grains. After the exposure, the sample is developed, fixed and stained to visualize the silver grains. Applications and importance. Emulsion autoradiography is used to study the distribution of radioactive isotopes in biological samples such as cells, tissues and organs. This technique is important for understanding biological processes such as protein synthesis, DNA replication and cell proliferation. Next is the film autoradiography. Film autoradiography is based on the principle that when an radioactive isotope decays in a biological sample, it emits radiation that interacts with photographic film. Earlier, it was interacting with the photographic emulsion. Here, we are providing the photographic film. The interaction results in the formation of a black spot on the film, which could be visualized by the developing film. Parts and working. To perform film autoradiography, a biological sample is placed in contact with a photographic film. The sample is then exposed to a radioactive isotope which decays and emits radiation. The emitted radiation interacts with the film. The emitted radiation interacts with the film and then exposed to radioactive isotope which decays and emits radiation. Which decays and emits radiation. The emitted radiation interacts with the film, resulting in the formation of a black spot. After the exposure, the film is developed to visualize the black spot. Applications and importance. Film autoradiography is used to study the distribution of radioactive isotopes in biological samples such as DNA, RNA, and proteins. This technique is important for understanding the structure and function of biological molecules. In conclusion, autoradiography is a powerful technique that is used to visualize the distribution of radioactive isotopes in biological samples. Emulsion autoradiography and film autoradiography are two types of autoradiography that are commonly used in molecular biology, biochemistry, and medical research. This technique is important for understanding biological process and the structure and function of biological molecules.